Hi, I'm Florent Lassalle. I work at the Wellcome Sanger Institute in the group of Nick Thompson. And today my talk will be about the background of the biology and microbiology of cholera and on the methodology that can be used to conduct genomic epidemiology studies of cholera outbreaks. And so this will be followed by a second part of the talk that is more specifically about the cholera outbreak that takes place in Yemen. So cholera, for those who don't know, it's so cholera, once an individual is infected, most individuals will actually go in asymptomatic, so carry the bacterium without developing symptoms. However, 20% of infected people will develop acute watery diarrhea, so AWD. And out of these, 10 to 20% will develop severe symptoms and risk severe dehydration. And this, if untreated, will lead to death in up to half of those severe cases. Thankfully, the treatment by rehydration therapy is straightforward and prevents death in more than 99% of cases. So this is done by giving either oral rehydration salts, so that's glucose with some electrolytes. That's for most cases. However, for severe cases, you can't stomach the fluids, IV fluids are given. And however, there is still scope for the use of antibiotics, and this is typically like to manage cases in outbreak settings. So this is to firstly limit the duration of the disease and manage the case of a specific patient, and also to limit transmission by limiting the number of bacteria that are shed in the stool of the patient, and then therefore less bacteria that can be transmitted to the next person. Vibrio cholerae is the etiological agent of cholera. It's a gamma proteobacterium, and it's flagellated. And it's got notably like a lipopolysaccharide coat with an O antigen, which is used, which type is often used like to discriminate different types of the bacterium. And its virulence that leads to the acute watery diarrhea, that is cholera, is related to the expression of specific virulence factors. So firstly, you've got the toxin co-regulated pilus that is thought to be required for the attachment to the intestinal wall. And then you've got the CTX toxin, which is the one that leads to the excretion of all salts, chlorine, potassium, sodium, and so on, from the intestinal cell into the intestine lumen, which leads by osmosis to the loss of all waters and salt, and like that can lead quite rapidly like to loss of all waters in the body. And so, importantly, not all Vibrio cholerae cells carry those genes in their genome. So that's why like genuine cholera is only caused by those Vibrio cholerae that carry the genes that code for the TCP and the CTX toxin. And so that is reflected by the definition of a confirmed cholera case by the Global Task Force for Cholera Control, which is that a suspected case has Vibrio cholerae bacterium of the serogroup O1 or 139 confirmed by culture or PCR. And in countries where cholera is not present or has been eliminated, that this strain should be demonstrated to be toxigenic, i.e. to carry the CTX toxin. Vibrio cholerae as a species is really diverse. And so we can see in its phylogeny. So the phylogeny is the genealogical tree of the species, so to say, and that phylogeny has many diverse lineages. However, the etiological agent of cholera is restricted to only a few of those lineages, and specifically to the clade, so like the group of branches and lineages that is called the pandemic generating clade, or PG clade, and that we can see here circled with red dashed lines. And so this clade is where most of the strains that produce the CTX toxin can be found. And interestingly, this correlates strongly with the distribution of serogroup 01 or 0139 strains. So most of the strains in this clade are 01 or 0139, and most of the 01 strains in the species are found in this clade. And that is why quite often the O1 type of the lipopolysaccharide O antigen is used as a shorthand to mean toxigenic Vibrio cholerae. But really, like, what we are looking for is like the fact that the strains produce the CTX toxin. And so within the pandemic generating clade, there is one specific lineage that's called the 7th pandemic LTOR, or 7PET in short. So that lineage is named after the 7th pandemic of cholera that has started in the 1960s and is still going on. And so this specific lineage is the only one that is currently causing epidemics and is being transmitted worldwide. However, there are many other lineages in this species that can also cause diarrheal disease, even though they are not genuine cholera, and don't lead to epidemics. And so that's because they don't have the same virulence factors, and so they only lead to sporadic infections, and quite often they're referred to as being endemic strains, which somehow refers to the fact that they are often picked up from the environment as one-off infections. And so if we want to study the infectious agents of those diarrheal diseases, we need to try and distinguish the different lineages that are observed of the pathogen. And so for that, like the best way is to look at to distinguish the best way to distinguish these lineages is to look at their genetic information, so their genome. So in the case of Vibrio cholerae, the genome is composed of 2 chromosomes, one large chromosome of almost 3 megabases, so 3 million base pairs, and one smaller one of around 1 megabase pair. And so depending on the lineages and the strains, you can have up to from 4,000 up to 6,000 genes encoded in the genome. And so specifically, like the 7PET isolates have around 4,000 genes. And so most of those 4,000 genes are conserved among the 7PET isolates, which means that most isolates of that specific lineage have the same set of genes. And only they can be differing slightly by what we call point mutations, so some changes in the nucleotidic sequence of those genes. And so those small changes in genes that are otherwise shared among strains is what we're going to refer to as core gene variation. However, when we compare strains that are more diverse, so that are separated by more evolution, so for instance from the 7PET lineage and another lineage, we will observe a difference in gene content in the genome. So there are genes that are present in the 7' strain and absent in the other lineage, and conversely. And so this type of variation is referred to as accessory gene variation. And so here we have the basis for two approaches that we're going to use to study the diversity of the Vibrio cholerae species. One is to study endemic Vibrio cholerae strains, where we compare fairly distantly related strains, which diversity can span the whole species. And the second approach, is to study groups of closely related isolates of Vibrio cholerae, and that's often for epidemic strains typically collected from the same outbreak, where they will be like clonally related. So in the first approach, where we study diverse and so quite often endemic, involving endemic Vibrio cholerae strains, this approach will take several steps. So I'm going to walk you through those steps. So first, we will conduct a de novo genome assembly. So for this, we first need to sequence the genome of the strain. So for this, we can typically use one of the Illumina sequencing platforms. So at the Sanger Institute nowadays, we use the NovaSeq. And so those produce short sequencing reads. So if you represent yourself like this blue line to be the genome, so the chromosome of the bacterium, that's put linearly, only small fraction of it will be sequenced at once. So this is like long of several million base pairs, and so those short reads are only up to 150 base pairs for the latest Illumina sequencers, and so they're actually like sequenced in pairs. So we call them paired end reads, where those 150 base pairs are sequenced in pairs that are separated by 1 or 2 kilobases of sequence. And so the idea is that we will sequence a lot of those short reads, like millions or billions even, so that to do like a dense tiling of the chromosome sequence. And so then what we obtain is something like small pieces of a massive jigsaw puzzle. And so the de novo genome assembly is the task of trying to put together those pieces of the jigsaw puzzle. And so for this we use bioinformatic tools and algorithms that will rely on the overlap of those small sequences to try and assemble them, and so relying on their homology. And so by finding bits of sequence that are shared between those reads, they're going to slowly build up like larger bits of sequence, and so those aggregated bits of sequence are called contigs, or contiguous sequences. And then relying on the fact that we have paired end information, where we've got these gaps that's not sequenced, but we know that those were near each other by a defined length in the genome, then we can build scaffolds. And so using this scaffolding principle, ultimately with enough coverage of the sequence, we can end up having a almost finished representation of the genome. And so that will be practically, in real terms, a file with lots of A's and C's and G's and T's, and that's the sequence representation of the genome. And so then, what to do with this nucleotide sequence of the genome? Well, the next step is to annotate it, which means to identify genes from the nucleotide sequence. And so by genes, I mostly mean protein coding genes, even though it could also be genes that code for RNA that have a particular function. And so here you have a screenshot of the software Artemis, that is a genome annotation tool, and where you can see the 6 different frames of the genome sequence. So that means the different reading frames in which the nucleotide sequence can be translated into proteins, and where so different frames have different genes that can be seen here as blue arrows. And really, like, annotation corresponds to identifying the begin and end of those genes, but also to predict the molecular function of those genes or the proteins that they encode once we have found their coordinates. And so here we have like a fraction of the plasmid, we'll see later, with different genes, and we can see notably highlighted here in black a sulfonamide resistance genes that is also framed in black in the middle of the top plot. And so this is just a different representation of the same bit of sequence, where I have put on a single line the different genes that are present in the different frames. So here we can see those in forward and reverse orientations, and also in different colors we've got different type of molecular functions. So like those in green are insertion sequences and different mobile transposases, and in red are those that are coding for antibiotic resistant genes. And so we also have the detail of the different molecular functions, and so we've got, for instance, an ABC transporter, that's a transmembrane transporter. We've got a transcriptional regulator, so that regulates the transcription into RNA of some genes. And then you've got hypothetical protein, which is a fairly common annotation to see in bacterial genomes, sometimes like in more than half the genes, which means that like we don't know what it does, what the molecular function is. Once we have identified the genes and their function, we can compare the gene content of different genomes. That's what we call a pangenome analysis. And that basically consists of classifying different types of genes into gene families. So those will be like genes that are similar, or we say homologous, across genomes, and see if those gene families are present or absent in the genomes. And so that's what we visualize here in this diagram, where columns of that heat map are different gene families, and rows are different genomes, and we can see so different genes being present in different genomes. From there, we can identify different compartments of the pangenome. So on one hand, we've got the core genome, which contains core genes or core gene families. So core genes are those genes that are present in all genomes in the dataset. So that if it's a strict definition of core genome, it can be also like a more flexible definition of the core genome, where we're considering genes present in at least 95% of all genomes, for instance. But here, like for example, it's present in all genomes. So those are framed in red in our example heat map. And then the other compartment is the accessory genome, containing accessory genes. And so those accessory genes are those which presence is variable across genomes. And so, based on that, we can use the core genes to infer a phylogeny of the organisms. So, for the detail of this approach, I will refer you to the presentation by Alice, which covers this in more details, but in summary, the approach is that like we take the alignments of all core gene families and we concatenate them and use that big alignment to infer a phylogeny. And so the benefit of this approach is that it's applicable to any set of genomes or isolates, whatever their diversity will always find a set of genes that is shared by all genomes and that we can concatenate and make a tree out of that alignment. The downside of that approach is that this tree will only be based on a fraction of the genome, which is that core genome fraction. And the problem is that this fraction may be quite small, because the more diverse the genomes in the dataset, the less likely they will share genes, so the less genes they will share. And so every time you include a genome that's quite distant evolutionarily from other genomes, well, they'll have less gene in common, and therefore the core genome fraction will be reduced, and therefore the alignment of core genes will be smaller and bear less information, and so that impacts the resolution of the tree that's inferred. And so here we have a trade-off between the size of the dataset we consider versus the resolution of the tree we infer. And what we can do next, once we have the tree, is use that phylogeny to study the distribution of the accessory genome in the context of the phylogeny. And so here we can see that there are groups of accessory genes which distribution matches some clades of the phylogeny. And so we can interpret this as genes that are associated to those clonal lineages, and really like the most parsimonious interpretation is that those genes have been acquired by the common ancestor of those lineages. And so those are lineage-specific genes. And they may be encoding specific functions that define the phenotypic specificities of those lineages. On the other hand, we've got genes which distribution doesn't match the pattern of the tree and are present on different lineages. And so those genes are likely to have been acquired by either of the lineage by horizontal gene transfer, because they were not present in the ancestor of those two lineages, because otherwise they would have been present in all genomes. Instead, like they must have been present in one of the lineage, and then maybe via conjugation on a plasmid, maybe via transduction through a phage, and so on, those genes have been passed from one genome to the other genome. And so that's what we call horizontal gene transfer, or HGT. And so a good example of this approach is the work that has been done to compare the closely related species Vibrio cholerae and Vibrio methecus, where they compared the gene content in a phylogenetic framework. So in this particular figure, actually, like they compare the gene contents of the various strains in their dataset to a reference genome, which is like one Vibrio cholerae 7-pet strain. And so they show like how much the gene content is conserved across isolates with respect to this particular reference strain. Another way of doing this comparison of gene contents is to do it properly in a phylogenetic context, so similarly to what I've introduced in terms of methodologies and what Alice has introduced as well in the previous talk. And so here in that paper, the authors have compared different isolates of different Vibrio species, including Vibrio cholerae. So isolates of the Vibrio cholerae species are together in the flat clade at the bottom of the tree. And so on the right-hand side, you've got this matrix of presence and absence of accessory genes, and here with a focus on virulence and antimicrobial resistant genes, and so showing the diversity of occurrence of those virulence and resistant genes across species, but also within Vibrio cholerae. The second approach is to study epidemic Vibrio cholerae strains, so that are expected to be very closely related, and typically when they come from a single outbreak. So in this case, we will align complete genomes. So we still start by sequencing the genome using typically short reads, but there we will aim to look at every bit of variation in that genome. And so what we want is to replace the position of those short reads in the original genome, not trying to piece them together, but rather like to put them on a template drawing. It's like if you had the template full drawing of the jigsaw puzzle and you're just putting the pieces of the puzzle in front of the template. And so of course we don't know the complete genome sequence of that isolate that we may just have sampled, but we can use a reference genome of a strain that is thought to be fairly closely related, and so to use that as a reference on which we will map the short read. So first, we will map the reads to the reference genome. So that means like for each sequence dataset we obtain for each isolate, we take, considering like individual sequence reads, we take those reads and we put them in front of the reference genome sequence, and so there are algorithms that will find where these align, and so the match doesn't have to be perfect, and that's because we can expect that there are mutations in the isolate genome that we sequence with respect to compared to the reference genome. But also there may be sequencing errors, so that when the sequencing experiment generates a mistake somehow in the sequence. But it's easy to tell them apart, and that's why, because here, for instance, we've got like 4 reads that are aligned to the reference, and you can see in red, you've got differences between the reads that are aligned and the reference. But those differences are consistent across the reads, so that means that it's truly there in the genome of the isolate we sequenced, and that's really a mutation compared to the reference. Whereas in blue, that C is only present in one read, suggesting that it's a random mistake of the sequencing experiment, and so that's discarded. So the next step is then to try and combine this information and to call variants, and so to build a consensus genome sequence from that variant calling process. So what we obtain is that a genome sequence that is very close to the reference, but for a few mutations that we inferred from the sequencing reads. And so then we can do that for several different isolates, and we'll see that like different isolates have different patterns of mutation, and so then like we can combine those sequences, and naturally that will make a whole genome alignment, because they're all aligned by construction, because they're based on the same reference, but they have different mutations, and so those mutations we call them single nucleotide polymorphisms, or SNP. And so those SNPs can then be extracted, like we don't need to look at the millions of base pairs that constitute a genome, like we can just look at those few differences that are observed across the isolates. And so here, like I removed like the few positions where we've got variation across isolates, and I reorder the isolates to show like their affinities. And so then that pattern of mutation can be used to infer a phylogenomic tree. And so here the advantage of this approach is that the tree is based on the variations that's observed over the whole genome. So that achieves the highest resolution possible. However, this assumes that all genomes share the same genes, so that like they can be fully aligned. So that requires that isolates are closely related. That works if they indeed come from a single outbreak, but then if there are mixed strains in the sample, then this assumption will be broken. And the risk is that like we've got strains that have different sets of accessory genes, and so those accessory genes in the strains that are not present in the reference won't be mapped, and so we're missing that aspect of the variation. So that's the trade-off between those two approaches. In this whole genome mapped approach, we discard the accessory genome variation, but in return we have a very precise and complete picture of the variation at the single base level. And so a good example of this approach was the study of the genomic history of the 7th pandemic of cholera in Africa that was led by François Xavier and many co-authors, including several from our team. And so it involved building a phylogenetic tree of more than 1,000 7-pet Vibrio cholerae genomes, and that includes 651 isolates taken from Africa over several decades, and also isolates of different global origins to provide context for those African isolates. And so, practically, short sequence based on that alignment, the maximum likelihood phylogeny was inferred, and so that's the tree you can see on the left. And what's interesting to do with this sort of tree is to then look at the clade structure, so how different isolates branch together, and compare that to the different geographical origins of the isolates. And so here you can see the branches of the tree are painted according to their geographical origin, and so brown is for Africa and green is for Asia. And so based on that you can see that like the African isolates always emerge from the diversity of Asian isolates, and so that's indicative of intercontinental transmission events or introduction into Africa. And almost, systematically, it would have been like from South Asia. And so those transmission events were numbered from T1 to T12 in that paper. And so further investigations can be conducted based on this sort of data, and that includes looking into the spatio-temporal spread of the pathogen. And so this relies on firstly like generating a dated phylogeny, so that's also called sometimes time tree. That's what you can see on the left. It's similar approach to what has been done previously, but also trying to calibrate the diversification events with real dates. So here, like relying on the fact that the times of isolation of those isolates was spread over several decades, and so giving like a good temporal signal to that phylogeny. And so here that tree then like depicts the different timings of diversification of the 7-pet Vibrio cholerae. And so then those timings can be related to the pattern of spatial transmission, and so that results into hypothetical but quite probable scenario of epidemic transmission with several events that were timed over the decades of the 20th century for transmission into Africa. So that's what's depicted on the right. And I believe François Xavier will give us like more detail on this study. And so to conclude, those two approaches that I outlined were used in the study of the Yemen cholera outbreak, and so that will be described in more details in the next talk. And so this is the end of this first part. Thank you for your time, and any question about this session may be asked during the question and answer session that will be scheduled later. And I let you move on to the next talk specifically about the description of our study on the cholera outbreak in Yemen. And with that, I want to thank the people in the team that helped me build this whole approach of investigation of genomic epidemiology of Vibrio cholerae, and my funding, which is Wellcome Trust. Thank you.